Hello, friends, welcome back to the biotechnology world. So, we were talking about biotechnology introduction. In the first lecture, we discussed the current progress and how it was working, what is the definition, and then how the current scenario is going on regarding biotechnological industry. So, but in this video, we are talking about historical background, like how it all begins and who were the pioneers of the biotechnology and how it all led to the latest technology of biotechnology, how it all worked. So we're going to discuss about all these stuff in this video. Let's start it. Antony van Leeuwenhoek. He was the first one. So let's talk about him. What was his servings and what was his services for biotechnology? It's the era of 1675. 1675 discovers bacteria using a simple microscope. So he's the person who first discovers the bacteria. So this was a great discovery at that time because bacteria are the smallest, living organisms rather than viruses, which are not termed as livings. The next was one Gregor Mendel. We all know the name of Mendel and Mendel's laws. He was an Australian monk who conducted the first genetical experiment using pea plants in the mid 1800s. So Mendel's law, we are all aware of Mendel's law of segregation and Mendel's law of independent assortments. These two were the major laws invented by Gregor Mendel. He was not actually a genetical scientist, but he just worked on it. He had around, I think, around 18,000 pea plants in his backyard, and he took researches on all type of pea, wrinkled, round, and everything. So it was a great serving from him, and we still follow Mendel's law of segregation and independent assortment. Biology is less in laws, but we still have laws from this person because he was fab. Often considered the founder of genetics. So this person is also known as the founder of genetics because he was the first one to make some genetical experiments. So now next is about Louis Pasteur. We are all aware of the term pasteurization. It was the time of 1870s. This proved the notion of spontaneous generation, describing the role of bacteria in spoilage and the scientific basis of fermentation. So he was the man, because initially it was thought that the bacteria the makes spontaneous generations and spoil the food that cannot be curable, and then we cannot preserve it. But he invented the process of pasteurization, fermentation, and he started to preserve the things for a longer period of time. So we still have preserved foods, but these are all basis of what Louis Pasteur invented in his 1870s. Created the rabies vaccine. So this is another big thing. He was the one to create the vaccine for rabies. Talk about Robert Hooke, 1665. Robert Hooke is well known for the invention of cell in the honeycomb structure, and he invented the first light microscope. First to observe cell in cork. Yeah. James Watson and Francis Crick. So, do you know about Watson-Crick model of DNA? I remember that. Yeah, they were the two who invented the Watson and Crick model of DNA. Actually, it was a double helical structure, and they observed it under the X-ray photograph in 1953. Paul Berg, 1972. Stanford University scientist who first developed recombinant DNA technology, a method for insertion of genetic material from the organism into another. So this was the beginning of the latest biotechnological industry when he developed the recombinant DNA technology by insertion of one segment. As I discussed in the previous first lecture, that we can insert one character in another organism of a character of our choice or our interest. So this was the basic of that, the recombinant DNA technology. We will discuss more about recombinant DNA technology because the whole biotechnology nowadays is based on this one. So Paul Berg was the founder of recombinant DNA technology. Okay, so it's about 1750 BC. Organism of technology emerged as the method of food production and plant and animal breeding. Use of bacteria to produce cheese, food preservation, use of natural enzymes in yogurt, use of yeast to produce bread, use of fermentation for producing wine and beers. This was the beginning of biotechnology by producing all these products by breeding plants and animals. In 1869, DNA is discovered in throat sperm by German Pasteur. 1990s, the word biotechnology is first used by Hungarian agricultural engineer. So we are progressing toward 1940s to 1950. Widespread work is undertaken to investigate the structure and function of DNA. This was the era when the work on DNA structure was started, and later on, when Mr. Watson and Crick invented the Watson-Crick double helical structure of the DNA, which is still acceptable with some more modifications. The U.S. Supreme Court approves the patenting of genetically altered organisms. Now, in 1980s, U.S. approved that you can use genetically altered organism as food products and much more. But we still have some problems using cloning methods by making different clones of the same thing or same organism. But the recombinant DNA technology is being used by and approved by Supreme Court of United States. A variant of GMOs and technology and biotechnology techniques are used in the field from agriculture to medicine. So, genetically modified and biotechnology techniques are introduced in the field for agriculture to medicine. Recombinant DNA technology extracts DNA from one organism. I have already told that what actually happens in the recombinant DNA technology, and represents specific improvement in the plant and animal. So they insert those DNAs, those segments, to improve the quality of plants and animals. The more crops they have, the more they will get. So they use recombinant DNA technology to get more crops out there, and while using plants, and more fertile, sometime mostly fertile. And if we talk about domestic, most milk producing animals, if we talk about recombinant DNA technology in the animals. Plant tissues culture gains widespread acceptance as a method to quickly and cheaply produce genetically identified plants. Tissue culturing is also a part of this biotechnology method. In 1990s, first transgenic organism, genetically modified, are introduced in the widespread agricultural production, particularly in the area of crops. BT corn, these are the examples. These are first transgenic organisms. 1990s, BT corn and soybean are introduced, offering natural insect resistance by the introduction of the gene from the bacterial Bacillus thuringiensis. In 1997, who knows about sheep? Dolly. We all know about Dolly. Dolly was produced. Dolly is the first animal clone for the diploid cell introduced in Scotland. We all know this Dolly. Late 19th and early 2000s, human cloning is outlawed in the US, and the first concerns over the use of human stem cells as research began to arise. So here comes the cloning methods is outlawed in the US. First concerns over the use of human stem cells in research began to arise. Here, the research on stem cells arrived. So, in the biotechnology agri sciences, we will discuss in later lectures. So it's done for today. Bye.